A pleasant afternoon to all of us. I hope all of us are safe from COVID-19 and from Typhoon Kinta. And now entering our par is Typhoon Rolly. We are nearing November already, a month before Christmas, and probably our schools have already taken off and the students have already adjusted to the new normal. But of course, we are hoping that the situation will normalize soon and we can go back to the usual face to face classes and we can interact with our teachers and classmates. Thank you for responding to our invitation to this webinar entitled Investigatory Project Ideas, Some Protocols in Plant Drug Research. This is the 14th webinar that we are offering since April when we had the first one. We will continue with this initiative until the end of the year with 3 more webinars coming up. So this online forum is being brought to you by Project STAR or Science Teacher Academy for the regions of the Science Education Institute, Department of Science and Technology. We're actually expecting a total of 100 science teachers, preferably teaching in the junior and senior high school, to join us in this seminar. There were a total of, I think, more than 200 plus who have signed up, but we're still waiting for them. So I hope that you find this forum valuable to you as a professional, especially those teaching investigatory projects, and hope that learnings may be applied to your online or face-to-face teaching. So stay tuned until the end of this webinar so you can answer the assessment questions we have prepared for you. Okay? So stick it out until the end. Once again, magandang hapon at mabuhay tayong lahat. So at this point, I would like to introduce our resource person. So we are happy to have with us for the second time our resource person, Professor Majal Rani Espiritu from St. Louis University of CAR to present to us investigatory project ideas, some protocols in plant drug research. Miss Espiritu is a university instructor, professor, supervisory instructor, but she's currently on leave right now at the St. Louis University. She is currently doing her PhD in botany at the University of the Philippines in Los Baños. She had her BS Biology at St. Louis University in 2015 and BS Biology also at the same university, St. Louis University, in 1996. Among the special training she attended are mostly from the Department of Science and Technology, SEI. One of which is the innovative teaching approaches for science and mathematics. Another one is on language strategies in science and mathematics. And recently, design thinking in K-3 science and mathematics teaching. Among her other accomplishments are the following. She's doing an ongoing research entitled Leaf Micromorphology of Vitex negundo at the UP Los Baños. And she has also been involved in various researches, too many to mention, at the St. Louis University. And she has been a Suke resource person in seminars and conferences, too many to mention again. And this one is the one I like most. She's our very own star trainer since 2016. So without further ado, it's a pleasure to introduce Maj Espiritu. Mamaj.
Hello po. Good afternoon po sa ating lahat. Kumusta po tayo? I hope we are all doing fine. We are all safe. Nako, despite this pandemic po, it's been a long time already. It's been months already and we would have expected it to only last for 2 weeks. 2 weeks. At siguro matagal na yung 3 months pero anong buwan na po tayo ngayon? Nako, pinakadepensa na lang po natin lalo't bumabagyo-bagyo is that magpalakas po tayo ng resistensya. Okay, so our discussion is on the investigatory project ideas, some protocols in plant drug research. Now, maybe you're wondering, bakit investigatory project agad? Hindi pa tayo bumabalik sa school. Now, teachers, ito na ang time natin para magplano. So, bibigay po natin ito para kahit papano we have already ideas on how to go about it. I would suggest that you make one umbrella study. Tapos kunwari kung meron kang 3 mga research groups, isang plant lang ang gamitin mo pero 3 magkakaibang protocols ang ipagawa mo sa kanila. Okay? Now again, why would you ask why would we talk about plants in the first place? Now we are all in our localities at kabisado natin ang kanya-kanyang mga plants. Po. So eto na po ang ating start ng webinar natin. So we were talking about plants and the availability of the plants in our own localities. So what available plants we have, we could actually study them. Um, Maybe you're asking, bakit hindi ang mag-aral niyan ang mga pharmacists at saka pharmaceutical companies? Unang-una po sa lahat, kadalasan ng mga pharmacists at saka pharmaceutical companies, sila na po yung nangre-refine, sila na po yung nagma-manufacture niyan. Pag-aaralan na po nila is yung mga chemical components. Pero when it comes to the plant itself, it is usually left to the botanists. And in the Philippines, there are only a few botanists. There are only a few. And madalas nga po hindi natin kabisado kung ano yung mga halaman natin sa paligid. Yun pala mga endemic po pala yan na nandito lang po sa Pilipinas mahahanapan. So the availability of the plants is one. Okay? Next one is the ease of propagation. Kasi ang mga plants po, pagka tinanim natin yan, mabilis na lang i-grow yung mga yan. Kung kailangan po natin ng additional na mga experimental studies, po pwede po natin siyang i-propagate ng ganun lang po kadali. Pangatlong reason po is yung ethical issues. Pagdating po kasi sa mga animal studies, pinakamahirap na part po niyan is yung pag-acquire ng iba-ibang mga permits para sa mga testing ng animals. Pagdating naman sa microbes, ang isang problema naman doon would actually be contamination issues. So, pinaka-feasible sa lahat, lalo pag ipapahandle natin sa ating mga students would be plants. Okay? And may I again stress that we have a lot of endemic plants in the Philippines because we are a mega diverse region, and considering that we are an archipelago, we are surrounded with water, may mga nag-grow sa atin dito na mga plants na talagang sa atin lang. Okay? Now maybe you're wondering again, why drug research? Now, magkadugtong actually yung reason niyan. Tumataas ang iba-ibang klase ng sakit habang bumababa ang biodiversity natin. What is the relationship? Pag tumataas po ang iba-ibang klaseng sakit, ibig sabihin this is already affected by climate change. Medyo na-modify po yung mga chemical makeup ng iba-ibang klaseng mga organisms, nag-mutate po sila, kaya mas nakakayanan po nila tayong i-infect. Now, on the other hand, ano ang mga pupwedeng gamot? Kadalasan po, ito nga po sa pandemyang ito, talagang obvious po na tayong mga Pilipino, talagang hindi po tayo prepared. Sa ibang bansa, kahit papano, meron po silang nakaredy na iba-ibang mga gamot nila. Pero sa atin po, wala po tayong prepared na maski na anong gamot. Kasi ito po ang isa sa kulang sa atin. Hindi po natin kabisado ang ating mga halaman. Now, another thing we have to remember, oh my gosh, we are presently experiencing the sixth mass extinction event. Hindi po natin yan nararamdaman. Pero marami po sa mga organisms natin ang unti-unti nang nawawala. Papaano po kung biglang nawala ang isang klaseng organism or ang isang klaseng plant? Tapos yun pala, siya ang pinakasagot sa isang klase ng sakit na kalalabas lang. So ito po siguro is a race against time. So since it's a race against time, then we teachers should already have an active role in this. So if we try to look at this process, this is the entire drug research and development process. So you would see that dito sa umpisa, basic research, dyan po tayo papasok. Dito po tayo sa basic research papasok. There would be thousands of different candidates na mga herbal plants dito. And then, dadaan po ito sa sangkaterbang proseso. Almost 10 years po ang pag-develop ng iisang klasing gamot. Yan po ay mananarrow down ng husto hanggang sa aapak ito sa clinical trials. 3 phases po ito. Kadalasan umaabot po ng 2 years ang isang phase. Tapos pagdating sa FDA review, tsaka na lang po matitira ang nag-iisang gamot na talagang pag-aaralan nila ng husto. So as teachers, kailangan po natin pumasok sa eksena. Because unang-una sa lahat, kabisado natin ang locality natin. Alam natin kung ano ang nag-grow na mga halaman. Alam natin kung ano yung mga available na mga trees, available na shrubs, available na herbs, even for weeds, even for the cryptogams, yung mga lower forms of plants, we have a lot of them and they are a variety. They could be sources. Now, likewise, as teachers and as science teachers, syempre iba ang credibility kasi natin. As science teachers kasi alam nilang talagang ang pinag-aaralan natin would have factual basis. So, in terms of impacting people, we are the ones who could actually impart better kung ano man yung natututunan nating knowledge with regards to this. And of course, lastly, we science teachers are the ones who understand ecological concepts and different environmental problems so that tayo ang makakapag-validate or makakapag-apply ng mga skills so that we are better to explore these plants, better to study the plants, better ones to utilize the plants or modify or design a way to use the plants. And definitely at the end of that, we would be the ones best to take care of these plants. Makakapagbigay tayo ng mga methods or mga designs kung paano aalagaan ang mga halaman na to. And with that, we target the two problems, the increasing disease and the decreasing biodiversity. Now, what is it in plants that we really study in terms of drugs? These are what we call as natural products. Lahat po ng living organisms may natural products. Pero syempre, since we are talking about plants, then we talk about the products of the plants, which are in the form of plant metabolites. So these are all products of their metabolism. So ang primary metabolism, yung simpleng breakdown and build up, would be giving rise to the products of carbohydrates, lipids, proteins, nucleic acids. Sa biochem, alam po natin ito, hindi po ba? Ito yung mga biomolecules natin. However, this would only be involved in growth. These are only involved in the growth and development of the plant. And alam natin as living organisms din, hindi po pwedeng growth and development din lang tayo. So, there is a secondary metabolism that happens. Usually, it makes use of carbohydrates, lipids, and proteins. gina-jumble-jumble yan pagdating sa metabolism. They put them together, pipilasin yung isa, idudugtong yung lipid, kundi yung protein, dudugtungan ng carbohydrates at saka lipids. And then, you come up with secondary metabolites that we have here as alkaloids, phenolics, steroids, essential oils, and lignin. Yung lignin, familiar tayo dyan, hindi po ba? This is the major composition of wood. Now, all of these secondary metabolites are not used for growth and development anymore, but they are used for defense purposes. What would be defenses against from? And this would be against UV radiation. This would be defenses against extreme salinity, extreme acidity, extreme temperatures, drought, extreme water, and even for parasites and disease-causing organisms, herbivores even. Yun ang mga gamit ng mga defenses na yan. So in the Philippines, we have the 10 herbal medicines that is approved and recommended by the Department of Health. Familiar po tayong lahat dito, hindi po ba? Yes, we all have this. Now, one thing that makes them really recommended by the Department of Health is that these plants have undergone several different studies already. Umabot na po na hindi lang yung halaman ng pinag-aaralan dyan. Na-extract na po nila yung mga active components niyan, na-elucidate na rin po nila ang chemistry ng mga yan, alam na rin po nila kung ano ang aapektuhan niyan o gagamutin niyan. Kaya po sila ang approved ng Department of Health. Kaya nga kung titignan po natin, maraming mga capsules tulad ng ampalaya capsule, bawang capsule, lagundi capsule, yung mga yan po, hindi po ba? So, eto po talagang trusted na po natin itong mga halaman na ito. Now, in terms of COVID treatment naman po, at the present time, there are two plants that are being studied. You have Coccus nucifera, okay, which would contain monolaurin. Ang mode of action po ng monolaurin is that it will target the lipid coat of the virus. So, parang egg po yan na pagka nasira niya yung shell, at least marirelease yung nandun sa loob, madali na lang pong mag-disintegrate. Ganun po ang mode of action ni monolaurin. Ganun din po pagdating kay Vitex nigundo, studied na rin po ito, being studied in the Philippines also. You have the presence of total phenolics, epicatechin, quercetin, catechin, and myricetin. Kombinasyon pa lang po nito ang pinag-aaralan at the present time. And most of the time, what they would be mentioning is that these compounds would be targeting the symptoms of COVID. May I stress on that, please? The symptoms of COVID lang, which would mostly be coughing, the difficulty of breathing, yun lang po. Pero kung ita-target niya yung virus mismo, so far wala pa naman po tayong lumalabas na study that would say so. Okay? Now, heads up lang po, we have here Ocimum sanctum and Artemisia annua. Ocimum sanctum is holy basil. And this is already, both of which are already being studied abroad. Kasi, pwede daw na malakas itong pangombat dun sa ating COVID virus. Artemisia annua po is damong Maria. Dito sa Pilipinas, I do not know. Pero since hindi pa naman din po ako pumapasok sa school, estudyante po ako ngayon, wala pa po akong naririnig na nag-aaral nito dito sa Pilipinas as of this moment. So if you would want also to start studying this, then you could. Okay, let's start with the research protocols. So our research protocols would be trichome and stomatal characterization, followed by fungal endophyte identification, and then we have brine shrimp lethality assay. Now for these research protocols, as you can see, then they would more or less use laboratory equipments. at the least, si trichome and stomatal characterization po gagamit ng microscope. Pero meron na rin po yung aspects ng stomatal characterization natin na medyo may introduce ako sa inyong software. So, it is best that before you choose the protocols that you're going to do, try to look at the community. Ano yung mga universities na nasa malapit sa atin? Ano yung mga gamit na meron sa kanila? Or kung mas malapit po tayo sa mga DOST regional offices natin, tignan po natin kung ano yung mga gamit na meron po sila sa mga laboratories nila. And then likewise, please try to gauge your students muna. Kasi some of these are easy, some of these are quite difficult, which is why sabi ko nga, this is recommended for those in grade 7 until grade 12. So siguro itong number 1 and number 3, pwedeng ipagawa po natin ito sa mga lower grades, 7 to 9 po siguro or 7 to 10. And then fungal identification would be a little bit complicated because I would be introducing to you a method of sequencing DNA. So may papakita akong software din sa inyo dito. So siguro mas magandang gawin ito ng grades 11 to 12. Okay? Now, another is, since we're talking about plants, before you start studying your plants, you please make sure that you contact a botanist so that merong proper identification yung plant ninyo. Kasi pinaka-importante po yun na before you do your studies, you should at least know the name of your plant. Okay? Aside from that, you please know its habitat, its characteristics, or kung paano man po siya nag-grow. Meron po ba ito sa bundok o dun lang po sa mga plains natin nag-grow? Because there are what we call as morphotypes. So it's best that we know our plant before starting with anything else. Okay. Now, yun nga lang po, teachers, I will not be able to include statistical analysis here anymore. So, ano na lang siguro, maybe you could ask for the help of statisticians that you know around o yung mga magagaling po sa math, baka pwede pong makatulong tayo, gagamit lang naman po tayo ng SPSS. Okay. So let's start with trichome and stomatal characterization. So what you see on the left side would be the hairs that are found on the leaves of plants. The very function of this would actually be protection. On the other hand, here at the right side, I hope you can see my cursor as I'm moving it. Here you would see the stomates, and they would still be found on the leaves of the plants. However, the function of this is more or less on transpiration. So binibigay ko na po yung bulk, yung pinaka puso ng magiging discussion ninyo pag ito ang inyong i-discuss. Okay? And then here, I have this here. This is a personal picture of mine in my study, Vitex negundo. This is accession number 59000. Ang lagundi po, iisang species lang, pero ang dami po niyang varieties. Um, I believe in the NPGRL, National Plant Genetic Research Lab, we have more than 40 plus 45 different morphotypes or different varieties of Vitex negundo. So, yun pong mga yun ay uumpisahan pa lang din pong pag-aralan. So, if you could just imagine, ang dami po nating pwedeng pag-aralan pagdating sa plants. So, ito po is just to introduce to you with two terms. This would be the surfaces of the leaves. Add eggshell with a D would be the leaf on the dorsal side. And as you can see, it's darker in color and it would have thicker cuticle. The ab eggshell side with a B naman would be the underside of the leaf. And as you can see, it's lighter in color and mas evident po ang ating mga veins. So in trichome characterization, this would be the materials that we would need. Of course, the leaf, you need clear nail polish, you need clear tape or scotch tape, and you need glass slides. end cover slips sometimes po, and of course the microscope. I hope we're all familiar in using the microscope kasi yan po talaga ang partner natin sa pag-aaral po ng mga living forms. So how do we acquire the epidermal sections? I give you two methods. The first one is the epidermal peel method. This is a method that works best when your leaf is thicker. Kumbaga, parang leaf ng duhat, leaf ng eucalyptus, leaf ng guava, yan po, yung medyo makakapal, na pagka sinubukan mo siyang i-break, mababreak siya. Pero you please do it slowly. Where do you do it? If you look at this picture, the break that you make should be at the region where it is closest to the midrib. Okay? And then, pagpilas mo ngayon sa kanya, your pilas or your peeling should be in the direction of the axial side or yung dun sa ating underside. Okay? So, eto, nakita natin. So, parang triangular shape po ito. Hindi po ba? Yan po ang gagamitin natin for studying. So, in a slide, mag-drop ka ng tubig. Ilagay mo yung ating natanggal dito. Okay? Lagay dito. And then you cover it with a cover slip. That is already ready for viewing. Okay? The next one, we have leaf impression method. Ito, madaling gawin ng mga bata ito. Ito kasing epidermal peel, kailangang teacher ang gumawa kasi kailangan ng fine motor skills. Pero dito sa leaf impression method, magagawa po ito ng mga bata. So you can see here, this is best for thinner leaves. Parang leaf ng lagundi, leaf ng saluyot, leaf ng ampalaya, yung mga ganun po kaninipis, yung hindi po natin mabibreak na leaf. So here you would have the leaf, kunwari, and then lalagyan po natin siya ng nail polish. And then you let it dry. Siguro mga 3 minutes po yan, 3 to 5 minutes. And then after which, kumuha ka ng scotch tape, pumutol kang scotch tape, ipatong mo. Okay? Pagpilas mo ng scotch tape, sasama ngayon yung nail polish. Idikit mo na ngayon yan dun sa slide. Hindi na po kailangan ng cover slip nito. Ready for viewing na siya. Okay? So, what do you expect to see if you would be studying trichomes? There would be two major types of trichomes. So this one is just one long horn-like structure na talagang ang tip niya is patulis. Ito naman, ang tip niya, meron tayong head. Okay? So this is the non-glandular trichome, yung walang head. Ang glandular trichome natin ay may head. Okay? Kasi dito naiipon yung kanyang secretions or yung kanyang products. So this is an example. Cannabis is, marijuana. Okay? So ang leaves ng marijuana, mayaman na mayaman sa glandular trichomes. Okay? So, you would have here a waxy layer that covers the head para hindi madaling kumawala or mabuhos yung secretion sa loob, which is contained in the secretory cavity. Now, those secretions are produced by the gland cells. The gland cells are here. Puti pero hindi masyadong kita kaya may diagram. Ito po ang itsura niya. Pag diagrammatic, ito ang itsura ng ating gland cells. Sila yung taga-produce ng secretion. And yung abscission layer, dito napipilas. So pag nirub mo ngayon yung surface ng leaf, dyan napupunit. Diyan kumakawala ngayon yung product o yung natural product na galing dito. So this would usually be in the form of tetracannabinoids. Ito po yung nakaka-high. Okay? Now, this would be the other types of trichomes na po pwede po nating i-observe under the microscope. And we could actually describe them to also be unicellular, single-celled, or multicellular. many-celled. So here you would see a unicellular type of trichome. Talagang makikita mong walang partition. Iisang cell lang siya. And this is the Malpighian type. Letter T siya when looking at the side. Pero when looking from above, mukha po siyang uod. Yun lang ang makikita mo. Hindi mo makikita yung stalk dito sa ilalim. And then we have the tapering type. One is uniserrate. Ibig sabihin, iisang sungay lang yan. And you have 1, 2, 3, 4 different cells. So this is multicellular uniserrate. Ito naman is multiserrate, multicellular. Okay? And then for the glandular type, we have two. We have pilate na mahaba ang stalk pero maliit ang head. And the capitate, which would have a small stalk and a big head. Next, we have stellate. So stellate is star-shaped. Sessile is immediately attached to the epidermal surface. While for the stalk, it would have a stalk tapos medyo floating siya. Okay? Meron siyang tangkay. Dendritic, finger-like ang structures niyan. At etong paborito kong magandang tignan, peltate. It's umbrella-like. Okay? I have included here a book. This is freely downloadable in Google. Ito na po ang link. Okay? So you could use that link already and then you could download that book. So this would be examples of trichomes. So we have here Eliaganus. Makikita po natin ang ganda ng itsura niya. Parang maliliit na, na bansot na mga payong na nandun sa epidermal surface. And we have Triumpheta. So this is star shape. Okay? Pelargonium has two types. One is glandular. Kitang-kita pa natin yung kanyang gland cells. And the other type is uniserrate. And for special mention, sila Portea, hindi po ordinaryong trichome ito. We call them as stinging hairs. So Laportea is lipang aso. Pagka natusok ka nito, nababreak yung kanyang tip, tapos magre-release siya ng calcium oxalate, yun po yung makate. Di po ba? Okay. Let's go to the next one, stomatal characterization. So here, we would do a part which is similar to what we have done with the trichomes. However, we would also do a part which is a little more complicated than previously. So ganun din ang gamit natin. You have leaf, clear nail polish, the tape, the slides, and the microscope. Idadagdag po natin ang ocular micrometer. Ito po siya. When you place this into your microscope and you look into it, you would see graticules. Parang may maliit na ruler doon. And this is something that you would need to calibrate kasi nagbabago po yan. Kasi syempre lumalaki po ang field of view pag ay lumiliit ang field of view sa bawat lipat natin ng objective. So ang pang-calibrate po natin dyan ay stage micrometer. So the stage micrometer is actually a slide. But at the center of that, you would see again a very small ruler there, which would be in micrometers. At ito po ay isasalpak natin dito sa ating microscope habang nakasilip ka sa ocular meter, and that is when you would calibrate. Okay? And of course, we have an additional, the Image J software. So what are we going to study with regards to stomata? So morphological muna yung una, tulad ng sinabi natin. And yung pangalawa would be this one, stomatal density, index, and size. And all of which would be discussed in terms of the transpiration of your particular plant. Okay, so let's just review what is the stomata. So the stomata is an opening in the underside of the leaf, most often on the underside of the leaf, and it would be guarded by the guard cells. And the guard cells would have vacuoles, na pagka ang vacuoles natin puno ng tubig, then uh, matigas ang ating mga guard cells, so they would be standing up, thus closing the stomatal pore. However, if the vacuoles do not contain much water, then the guard cells are a little bit flexible. So since they are flexible, the tendency for them is to bend, opening the stomatal pore. Okay? Now, how do we characterize this entire apparatus? We characterize that in terms of the subsidiary cells, which would be the cells that are immediately surrounding the stomata. Okay? And likewise, i-compare din natin yan sa ating mga epidermal cells, which would be the rest of the cells in that particular layer. Okay. So ito din ang gagamitin natin na pag-acquire ng ating study material. Epidermal peel method pa rin at saka leaf impression method pa din. Okay? So what do you expect to see in terms of morphological characterization? These are already the types. So you have the first one to be anomocytic or irregular celled or ranunculaceous. Bakit? Kasi nahahanapan po siya sa mga ranunculaceae. This is a family of plants. Okay? Now, how are they described? You have this to be a limited number of subsidiary cells. In this case, in the diagram, that's 1, 2, 3, 4. Immediately surrounding the stomata, there would be 4 subsidiary cells. And here it is mentioned which are quite alike the remaining epidermal cells. So kung irregular ang shape ng mga ito, most likely yung epidermal cells sa paligid ay irregular din ang shape. But for this example that we have, Tradescantia species, then you would see that the subsidiary cell is actually the same as that of the other epidermal cells. Di po ba? So that is anomocytic. Next, we have anisocytic or unequal celled. This we find in crucifers. Okay, you have 3 subsidiary cells, 1, 2, and 3, where one is distinctly smaller than the other two. This is smaller than the other two. So in our example, Solanum species, you have 1, 2, 3 subsidiary cells, and this would be the smallest among the 3. That is anisocytic. Next, we have parasitic or parallel-celled. We find this in Rubiaceae, kaya siya rubiaceous din. So, parallel sa longitudinal axis ng pore at ng guard cells. So, ito ang pore, ito ang guard cells, ito ang ating subsidiary cells. But look, the subsidiary cells are smaller here, while for here, the subsidiary cells are much, much bigger than our guard cells. Okay? So an example here is Ixora. So as you can see, ito yung kanyang subsidiary cell, malayong mas malaki kesa dun sa ating guard cells. Okay. Next, we go to diacitic or cross-celled. From the term cross-celled, alam na natin yan, hindi siya parallel. Kabaliktaran nung parasitic. And we find this in Caryophyllus C. Okay? So this would be the guard cells at ito ang ating subsidiary cells nakahalang. Kaya siya diacitic. Now, an example given here is dianthus. So this would be our guard cells. Nakahalang sa guard cells ang ating subsidiary cells. Okay? So kabaliktaran ng parasitic. And then we have actinocytic. So the stomata are surrounded by 4 or more subsidiary cells. That's 1, 2, 3, 4, 5, 6, 7, 8, 9 subsidiary cells. And they are elongated radially to the stomata. Parang nakatayo siya, pero yung paa niya nakadikit dun sa ating guard cell. Okay? Do you see that? Nakatayo sila pa ganon. So they are elongated radially. Now similarly, for Jatropha, ito po ang makikita natin. So you have the subsidiary cells to be elongated radially. In contrast, we have, cyclocytic. Again, 4 or more subsidiary cells siya. Ito actually lahat subsidiary cells niya yan. Oh. Okay? But they are arranged in a narrow ring around the stomata. So they would be narrowly arranged, lahat sila nakahiga dun sa ating guard cell. Okay? So an example is your sile or your piper species. So this is our guard cells here. Tapos naka-arrange. May maliit, may malaki, pero naka-arrange sila paikot at nakahiga dun sa ating mga guard cells. And the last is gramineous. This is what we find in grasses. Okay? Madali lang i-identify to kasi dumbbell shape agad ang ating mga guard cells. If you look at the guard cells of the others, they would be bean shape. Pero if you look at the gramineous, they would be dumbbell shape. Okay? This is in Poaceae or in grasses. This particular example is actually in Zea maize or maize, corn. Okay? So that is for morphological characterization. Now, what is the relevance of morphological characterization of both trichomes and stomata? Sa taxonomy po yan. Importante po kasi ang taxonomy. Actually, in pharmaceutical biology, they are not focused on the ordinary taxonomy anymore. Pero they are more focused on chemotaxonomy already. That in certain types of families or even genus of plants, meron silang very specific na compound na sila lang talaga ang nagpo-produce. Kaya napaka-importante pa rin po ng taxonomy pagdating sa drug research. Okay, let's look at the others. We have stomatal density, index, and size. Now, this is my recommendation. My recommendation. Pero if you have a better microscope, then go for it. Gamitin ang mas mataas na magnification. So, for the eyepiece or the ocular magnification, I suggest you make use of 10x. Okay? 10x po. So that you have the total magnifications to be 40x for LPO, 100x for MPO, 400x for HPO, and 1000x for oil immersion objective. The oil immersion objective, take note, we're going to use an oil here. Ipapatak mo siya sa ibabaw ng cover slip. So that pag nilagay mo yung objective doon, mas lalo pa siyang mamamagnify kasi magre-refract ngayon yung daan ng ilaw dun sa ating oil. Okay? Now, for the uses of these ones, use the low power objective for survey only. Huwag ka munang magpipicture. Okay? By the way, teachers, from the trichomes until dito sa stomata, wag po kayo hangga't maaari para hindi aksaya sa oras. Wag po kayong mag-observe at mag-analyze agad habang nandun kayo sa microscope. It's best that you already take a very good picture and then analyze it after. Okay? So kung gagamit kayo ng microscope, siguro in just one day marami ka nang napasadahan. Okay. For stomatal density, you could already make use of middle power objective. And for stomatal density or index, po pwede na sa high power objective. Pero pag stomatal size ang i-measure natin, then it's best that we make use of the oil immersion objective. Okay. So for stomatal density, index, and size, kailangan po nating mag-calibrate ng ocular and stage micrometers. Okay? Hindi ko na po yan mapapakita sa inyo dahil wala po akong microscope dito sa bahay. So nagbigay na po ako ng links dito so that you could already watch the tutorial, some of which would be 15 minutes long, some would be 20 minutes long, but then I recommend this too. Okay? Sinama ko na rin po dito yung pag-compute natin ng field of view because this is a very important aspect for your density and index, yung field of view natin. So the formula is similar to that of measuring the area of a circle. So you have 2 pi r. Okay? So, saan tayo kukuha ng diameter of the field? Pag na-calibrate na natin ang ocular at saka ang ocular micrometer natin, that's the time that you measure the field of view. So, dulo hanggang sa kabilang dulo, you measure that, and that would be the diameter of your field. Divide that into 2, then you get the radius. Take note, you take pictures and analyze after. Okay? And then we have stomatal density. Simple lang ang stomatal density. You just count the number of stomata in that particular field of view. This is my picture for Vitex Negundo. This is the number of stomata in one field of view. How many? You have 5. And my field of view, according to my calibration, is 260 micrometers. So I have 5 stomates in a 260 micrometer field of view. Okay? Pero, syempre, hindi po pwedeng iisa lang ang iti-take mo. For every specimen, you have to take at least 20 field of views. So, if, may I just go back? Sorry. So, if this would be your field of view, okay? This would be your specimen. Then, tingin ka dito ng isa, kunan mong picture, and then isa ulit dito sa baba, kunan mong picture, dito sa baba, picture ulit, dito sa baba, picture ulit. So, you more or less take 20 pictures of that entire specimen. So, sa isang specimen, you should have at least 20. Okay? And then you get the average of that. So, yun na ang stomatal density ng buong plant natin. Okay? Next, we have stomatal index. Dito, you're not just going to count the number of stomata, but you're also going to count the number of epidermal cells. So, number of stomata over number of stomata plus number of epidermal cells times 100. So, ang stomata natin is 5, 1, 2, 3, 4, 5. And then we have the epidermal cells to be 20. Okay? So that is 5 over 5 plus 20 times 100. We come up with an index of 20. But then again, take note, you should at least make use of 20 fields of views per specimen. Okay? Para makuha ang average. This is an article I have included in this presentation that you could model after, or you can pattern your study on this type of study. So this is by VJ Paul. Siya din po ang ginamit namin nung unang nag-study kami ng stomata. Sa kanya po namin pinattern. Kaya sa kanya po kami nakapag-aral ng maigi. Okay. And then we go to stomatal size. Now, bakit mas komplikado ang stomatal size? Kasi kailangan mong tignan yung isang buong stomata. Okay? So you're going to measure the length in the blue line and the width in the red line. Okay? And this would be 20 stomata per specimen at least. Okay? Parang ang hirap naman po niyan, ma'am. Actually, once you have taken a picture in, let's say, HPO or oil immersion, po pwede na ninyong i-load ito dito sa image J. I have included the sites where you could freely download image J. Dalawang sites po ito. Okay? So, eto naman po ang mga tutorials din kasi hindi ko po maituturo ng todo-todo pero ipapakita ko lang po ng konti. Ang unang gagawin po natin before we could make use of the image J is to set scale. And then that's the time that you measure the size and you measure the area. I would shift to my image J. Okay? Sorry po. Ito po yung working ha, this one. Ito po ang nandun sa slide. So for image J, papasok ka sa analyze, andito na po ang set scale. Okay? So when you set scale, you are going to make use of a picture. Okay? You should have look for pictures in various articles that would have the same, the same magnification as what you have used. Okay? Ano ang pinaka-important aspect dyan? Ito po, nakikita nyo po ba yung 5 micrometers dito sa baba? Gagamitin po natin yan. Kasi ang pag-analyze po ni image J is per pixel. Pero ang pag-measure niya ng per pixel is nakadepende dito sa 5 micrometers na to. So pag sinalpak nyo po ito sa image J, you are going to measure one end of this papunta sa kabilang end nito. Okay? Ire-record na niya yan kung ilang pixels ang equivalent sa 5 micrometers. Ganun po kagaling si image J. Okay? And then, after which, pwede na po tayong, sorry po, pwede na po tayong mag-load ng ating picture. So, I have one picture here. Okay? Ito na po siya. Okay? So, let's say, kung ito siya, i-click nyo lang po. Most of the time, you'll just click this up to here. Okay? Na-measure na po yan ni Image J. Tapos, meron na po siyang process dito. Siya na po ang magre-record. Once you are done measuring 20, so i-measure mo din ito. I-measure mo din yan. I-measure mo din ito. So, parang nakailan na po tayo? 4 na po yun. I-record na po yun ni Image J. And then after po nun, maglalagay na rin po siya ng average. Okay? Ganun po kagaling si Image J. Kaya gusto ko po talagang hanggat maaari, ma-introduce po kayo kay Image J. Kasi kasing dali lang po ito ng paggamit ng paint. And you could already analyze not just stomates but also microorganisms even. Even for your trichomes po, pwede po siyang i-analyze making use of image J. Okay? Pakipanood na lang din po yung mga tutorials na ito. Sinama ko po talaga dito sa presentation para pwede nyo pong ulit-ulitin. Balik-balikan. Okay, let's move on po. In fungal endophytes, so you know the term fungi. Endo means within, phytes means plants. So these are fungi that live within plants. Okay? Now, they would be living in such a way that they live talaga within the cells even. So this is one fungal hyphae that crosses, tumatawid pa nga siya sa ibang cells ng plant eh. Ganyan si fungal endophyte. Meron din po tayong bacterial endophyte, pero fungal endophyte po ang i-discuss natin ngayon. Okay? What is the relevance of this? I introduced to you Taxus brevifolia. This is a type of pine tree, a pinus, Pinaceae siya. Okay? And meron po siyang around 100 plus endophytes, fungal endophytes. And all of those are capable of producing a particular compound that we call as taxol, kasi coming from taxus, and you would have that to be released in the market as paclitaxel. And this is an anti-cancer drug. Okay? Now, here you see the relevance of the symbiosis because the fungus would definitely be dependent on the plant with regards to its nutrients. Tama po ba? Kaya kung ano man ang mapuproduce ni fungus, kailangan talagang nandun din sa chemistry ng plant. So dito tayo papasok muna sa pag-aral kay fungus. Pagka higher or kung gusto niyo dagdagan yung research ninyo, then pag-aralan niyo rin yung plant. Pero ang chemical elucidation would actually be needing a lot of laboratory instrumentation na medyo mahirap hanapin. Kaya ang gumagawa po kadalasan yan is yung may mga gamit na mga scientists. Okay? So doon po muna tayo sa preliminary protocols po tayo. Okay, so the methodology for fungal endophytes would comprise of 4 different stages. Medyo madugo po ito, kaya sabi ko nga po, mas magandang ipagawa ito siguro grade 11 and grade 12. Okay? Pero talagang close supervision po kayo dito. So you have media preparation, explant preparation or surface sterilization. You have crude culture and subculture, and you have identification. Please take note that you have to prevent contamination and infection at all times. So, ibig sabihin, hindi po pwedeng nagdadaldalan, hindi po pwedeng hindi nakamask. So hanggat maaari, wear your gloves, wear your lab gown, wear your hairnet, wear your mask. Okay? For asepsis. Kailangan po kasing ulit-ulitin yan sa mga bata pagka yung babantayan sila sa laboratory. Hindi po ba? Okay, so let's start with media preparation. Media preparation po, magluluto tayo ng gelatin. It's actually scientific lang po yung media preparation, pero magluluto tayo ng gelatin. This is a special type of gelatin. We call as sabrodextrose agar. And this is the most appropriate agar for growing fungi. Okay? Now, all of the nutrients in the sabrodextrose agar is talagang ito yung mga nutrients na required para mag-grow ng fungi. Okay? Now, before we make our media, we already try to compute how many Petri dishes we're going to use. Okay? So if you think you're going to use 10 Petri dishes, then siguro ang kakailanganin mo is around 200 ml of the media. So how do you do that? Parang magluluto talaga ng gelatin. Pero ang gagamitin natin dito ay distilled water po. May I stress on that? Distilled water. Not chlorinated, as much as possible, not tap water. Because there would be some minerals that would be present in the water na po pwedeng maging mali ang kalalabasan ng ating experimentation. So please make use of distilled water. 250 ml, and then you get 7.5 grams of the sabrodextrose agar. Ilagay mo siya doon, and then iinit mo siya making use of a hot plate. Okay? I would suggest you make use of an Erlenmeyer flask. Bakit? Kasi kailangan po nating takpan ito after. So pag na-melt na lahat yung ating gelatin, yung ating agar, melted na siya. Please do not make it boil. Okay? So pagka fully melted na siya, talagang incorporated na siya, homogeneous na ang ating solution, then you cover it with a cotton plug. Then after which we're going to sterilize that. So kadalasan, ito ang ginagamit. This is an autoclave. Isasalpak mo po siya dyan, tapos ipoprogram mo na siya for 121 degrees Celsius for 15 minutes. Bahala na po siya dyan. Pero actually, it's not just going to take 15 minutes. It's going to take about an hour. Kasi yung pagtaas po ng pressure at pagtaas ng temperature would take about mga 30 minutes na. And then 15 minutes po ito. And then another 15 minutes sa pagbaba po ng temperature at saka pressure. Tsaka mo siya po pwedeng buksan. Hindi po pwedeng buksan ang autoclave. The same way as you have your pressure cooker, ang pang-autoclave din po kasi nung iba is pressure cooker. Hindi po pwedeng bubuksan ang pressure cooker habang mainit pa or habang nandun pa yung pressure. You have to release it kundi sasabog po yan. Okay? So include with it already your another 200 ml of distilled water. We have to have sterilized distilled water. And you include the Petri dishes that you're going to use. And likewise, another Petri dish na gagamitin natin as sangkalan. Opo, dun po tayo magsaslice for the explant. Explant is a term that we use for the part of the plant that we're going to excise or cut for study. Okay? So, nag-autoclave po tayo, nag-sterilize, mainit pa siya, nilabas natin, we have to cool it down for at least umabot ng 45 to 50 degrees Celsius. Pero, wag na po sanang bababa sa temperature na ito kasi titigas na po siya. Okay? So, once you have come to this temperature, you start swirling. At least slowly lang po na swirl, and then that's the time that we add the antibiotic. What is the use of the antibiotic? Kasi if we do not place an antibiotic, there is a possibility that bacteria will grow. We do not like bacteria because this is a fungal culture. So I suggest make use of chloramphenicol, which would target both gram-positive and gram-negative bacteria. Okay? So kung gumawa ka ng 1 liter ng media mo, then 50 milligrams ang ilalagay mo. Okay? Dissolve that first in 10 ml of ethanol, and then isama mo siya, and then you keep swirling hanggang sa makita mong nag-dissolve na ang ating antibiotic. Okay? From there on, you dispense into Petri dishes. You could make use of a pipette pagka yung talagang tipid-tipid ang iyong media para eksaktong 20 to 25 ml lang ang nailalagay mo. Pag buhos-buhos system po kasi, baka masobrahan yung iba. And then you cover it and then allow it to solidify. Okay? So habang pinapasolidify mo ang iyong agar sa mga Petri dishes, pwede ka nang gumawa ng explant. So this is explant preparation or that is surface sterilization. You're going to use maliit lang na beaker, po pwede na. Okay, 100 ml lang yung nilagay ko dito. And you should have forceps to prevent the leaf from falling off pagka yung binubuhos mo yung solution. And of course, you have your explant. From here on, please be careful with your explant kasi pagka na-injure mo, kunwari yung plant, nasugat mo siya, then pupwedeng any of this pumasok sa loob. Mamamatay ang ating fungi. So pagka i-grow mo na siya ngayon sa media, walang mag-grow kasi na-sterilize pati yung loob ng plant. Okay? So prevent any wounding. So the first one that you do is to wash the leaf. If explant po pala ang ginamit kong term dito kasi po pwede po siyang leaf, po pwedeng root, po pwedeng stem. Okay? Kaya explant po ang ginamit natin. So you have to wash it with tap water and soap. Banlawan mo ng maigi. After which you rinse it 3 times with tap water still. So para matanggal yung mga minerals or anything chlorine in the tap water, we rinse it again with distilled water 3 times. Okay? So ibuhos-buhos lang to. Buhos-buhos system lang po siya. Pero naiiwan po ang ating leaf dun sa ilalim ng beaker. And then after the distilled water for 3 times, you immerse it in 70% ethanol for 1 minute. Sinubukan ko na po dito ang 70% isopropyl alcohol. Pwede po siya. Pwede po. So enough to cover lang po. Wag po yung pupunuin ninyo hanggang 100 ml. Enough to cover lang po yung ating leaf. And then after which, buhos mo yung ethanol, and then ang isunod mo naman ngayon is aqueous sodium hypochlorite. That is your Zonrox. Okay? So how do you make it 10%? That would be water, which is 9 parts, and your Zonrox, which is 1 part. 10% aqueous sodium hypochlorite na po yun. Okay? 5 minutes ninyo siya ibababad. And then after that, immerse it again. Buhos ulit siya and then immerse ulit sa 70% ethanol. 30 seconds po ulit yon. Okay? And then the last part, you remember yung distilled water na sinama po natin sa autoclave? Dito na po natin siya gagamitin. So we have to wash again for 3 times with the sterile distilled water. That is to remove any of this. Kasi pagka may natira po dito at in-slice po natin ang ating leaf, po pwedeng may makapasok po, wala po tayong mag-grow na fungi. Okay? So this is from the Protocol of Hyde and Soitong, 2008. And then, maalala nyo po yung inotoclave din natin na Petri dish. Gagamitin natin yan as sangkalan. And then we should make use of a forceps and a scalpel. And definitely, paulit-ulit na sterilization, kailangan natin ng denatured alcohol. Okay? So this would be your leaf. You cut it 1 centimeter by 1 centimeter sections. So isang section ito, isang section ito, isang section yan. Pero bago nyo i-cut, paki-sterilize po muna dito sa apoy natin. Okay? And pag na-cut po natin yan, ilagay po natin sa media ng ganyan, firmly. Place it on the media firmly. And then after which, you make sure that you seal it with tape. Okay? Seal it with tape para wala pong papasok na contamination. And then, you leave that for 7 to 10 days. Room temperature, please, hanggat maaari sa lugar na hindi naaarawan. kadalasan po ang pinaglalagyan ko nito pagka sa laboratories, nilalagay ko po sa loob ng cabinet kasi mas gusto po ng fungi na mag-grow sa mas madilim na lugar kaysa sa mas maliwanag. At mas gusto din po niya ng mas malamig ng konti at mas moist na lugar. Okay? Huwag po sa incubator ha, kasi ang pag-grow po ng bacteria is incubator ang gagamitin at 36 to 37 degrees Celsius. Pero pagka fungi po, dito lang po tayo sa temperature range na to, 23 to 25 degrees Celsius. And then you wait for the growth of the spores. Makikita nyo po yung growth ng spores pag eto na ang lumabas sa ating crude culture. Okay? Ito po ang kalalabasan niya. Now, I'm presenting to you two subculture methods. The first subculture method, this one, is best for DNA sequencing. Okay? So, ito ang ating ginawa. So, kunwari, yan po ang ating mga leaves. So, ano ang gagawin natin? This is one type. Another type, kapareho lang nito. And this is another type as well, kapareho lang nito. Okay, kapareho lang din yan. So we have three. So you get a Petri dish, a separate Petri dish, yung sterile at meron na rin siyang media. Kuha ka ng loop, kuha ka dito ng sample, and then ilagay mo dun sa media. Okay? Kung ilan ang growth mo dito, yun din ang number dapat ng plates mo. Okay? So kung meron kang 1, 2, 3, 4, 5, 20 na iba-ibang klaseng fungi, dapat 20 din ang Petri dishes mo na kalalabasan. Okay? So may kanya-kanya na silang bahay. Hindi yung tulad nito na nagsama-sama sila sa iisang bahay. May kanya-kanya na silang bahay dito, which is why we call it as subculturing. Okay? From here on, you could already have this for DNA sequencing. Okay? I suggest that you email this two websites. You have the Philippine Genome Center and Macrogen Korea. I'm not really updated with how much ang DNA sequencing nila. Pero nung last na ginawa ko po ito, nasa 300 to 350 pesos per sample po yun. Per sample. Okay? So, ang gagawin nyo po, email nyo sila and you make an appointment. Kung sa Philippine Genome Center po kayo, actually maganda. Kasi gagawa kayo ng appointment tapos tanungin na rin po ninyo kung gusto ninyong manood habang nag-extract sila ng DNA. Okay? Maganda pong experience yun. Panunuorin ninyo kung paano nila ipaprocess. Now, please make sure that you instruct them. They will not get the entire DNA sequence of the fungi. They are only going to get the ITS region. This is a segment of the DNA that encodes mostly for characteristics, certain characteristics of fungi. Okay, these are conserved characteristics of fungi found in this particular segment. Okay, I have included here as well. So, eto na po siya, no? Kunwari, nakuha ninyo yung DNA sequence. Ito po ang matatanggap ninyo. I-email nila agad dito sa inyo. Kaya in about 3 to 5 days, makukuha na ninyo agad ang results. Kung ayaw po ninyong sumama, okay lang naman din po. Pero i-email na lang po nila ito. Ito po ang example ng mga ipapadala nilang results sa inyo. This is actually the important one, the notepad. Okay? Kasi ito, mahirap pong input ito sa software. Ito po ang may input natin sa software. Okay? Sa notepad. Ito po yung yung sequence na po niya ito. Okay? So, after which, punta na tayo sa website na ito. This is NCBI. Gamit na gamit po namin ito for bioinformatics. Pag pumasok ka sa NCBI, may I just go to NCBI? Pag pumasok ka, eto po siya. Okay? So we click this. I hope gagana po siya. And then, eto po, andito po tayo sa enter accession number or FASTA sequence. You remember your notepad? Ito po ang sequence natin. So, Control A, Control C to highlight everything and copy. And then, Control V to paste it all there. Okay? And then after which, baba ka, wag mo nang pakialaman yung iba dito. Wag mo na siyang palitan. Pindutin mo na lang yung blast. Okay? And then you would see that you have a request ID here. Searching siya kasi lahat ng database papasadahan niya para basahin niya lahat ng sequences. And then voila, andito na po ang ating results. That again is your reference ID. And if you would see here, this is the important one. The percent identity, it's 100% for 10 different species, different strains. And ano po ang lumalabas na organism natin? It's Aspergillus niger. Okay? So you could just go to alignments. Yeah, okay na po siguro ito. Pwede nyo na lang pong kunan ng picture ito. Ito po ang accession number kung gusto nyo yung pinakauna. Okay? Ito na po yung ating result na isasama dun sa ating study. Okay? So let's proceed. Here you still have blast tutorials that I have included. May links pa rin po dito para po kung hindi ninyo masyadong naintindihan, pwede po ninyo siyang balik-balikan. Okay, this is another subculture method. This is the slide technique or the microculture technique. This naman is best for microscopic method of identification. Hinuli ko lang po ito in comparison to DNA sequencing because I would want to stress out that the identification using a microscopic method is still best para sa mga kabataan because they will learn how to make use of the dichotomous keys sa pag-identify ng mga organisms. Okay? So anong gagawin natin dito? Panibagong setup po actually siya. You would need a Petri dish. Lagyan natin ng filter paper sa loob. Tapos po pwede pong may patungan. Sa akin po, ang ginamit ko dyan is application sticks. Pero dito po sa picture, ang ginamit po niya is slide. Tapos magpapatong po siya ng isa pang slide sa ibabaw. Yun po ang i-autoclave. Lahat po yan i-autoclave. Pero pag natapos i-autoclave, tsaka po tayo magka-cut dun sa sterile na media. Maalala nyo po, meron tayong medium na ginawa. Kakat po natin yan ng ganito kaliliit, patong dito sa slide. And then after which, you get from the fungus, and then you inoculate here, 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 and here. Okay? So lahat yung mga sides na yan, dapat meron tayong ine-expect na mag-grow na fungi. And then you put on the cover slip. Okay? And then we already wait for 3 to 5 days. So after 3 to 5 days, yung cover slip lang po ang ating tatanggalin. May mga nakadikit na po na spores doon. Okay? Ilalagay lang po natin siya sa glass slide, ready for viewing na po under the microscope. Hindi po masyadong clear sa picture ko kasi in bulk ko po sila na picturean, pero I hope you can see this agar block. Tapos meron po siyang itim-itim. Yan po yung nag-grow. So tinanggal ko lang po yung cover slip dyan, and then I placed it on a slide, and this is how it looks like. Okay? This is cladosporium. Now, how do you identify this? I have given you here 4 different books that you could download freely from the internet. Ito po yung mga same books na ginamit ko para mag-identify po ng aking mga fungi, ng mga fungal endophytes ko. Okay? Last but not the least, we have brine shrimp or Artemia species, Artemia salina lethality assay. Brine shrimp ang pangalan niya, pero honestly po, hindi po siya shrimp. Mas kamag-anak po niya, mga water flea. Oo, maliit po siya. And initially, nahahanapan po siya sa mga um, salty lakes sa bandang Europe. Pero at the present time po, medyo naikalat na rin po sila. So the brine shrimp is able to thrive in very, very salty water. It is actually able to thrive in 50% concentration. So could you just imagine, isang basong tubig, tapos hahaluan mo ng kalahating basong asin. Tapos, i-dilute mo. Yun siya. Kaya niyang mabuhay doon. In addition to that, kaya din po niyang mabuhay sa potassium permanganate at saka silver nitrate. Ganon po katibay si brine shrimp, which is why they usually make use of the brine shrimp to indicate if there would be substances that are poisonous. Kaya ito po ginagamit sa mga toxicology studies, si brine shrimp assay, BSLA. Okay? And then, for other studies naman, they make use of the brine shrimp lethality assay for cytotoxicity testing. Ano naman yon? Ang implications ng cytotoxicity testing is meron kang substance, gusto mong i-test kung ito ay may effect sa isang particular type of cell. And in this case, ang ating cell types would be carcinoma cells or cancer cells. Okay? Now, you would see that in Purdue's Cancer Center, ito na po ang talagang ginagamit nila as pre-screen for 6 human tumor cell lines. Ibig sabihin kung pre-screen, yan yung pinakaunang screening system na gagamitin nila para i-identify kung may cancer ang isang tao. Okay? So, eto na po ang gagamitin nila. And then, pag nag-positive, tsaka po ito ipapasa sa iba pang klaseng mga assays. Okay? Kaya po siya high throughput, pero very simple po ang method nito. So the methodology would involve 3 major steps. The first one is the preparation of the plant extract. The second one is the hatching of the brine shrimp eggs. And the third is the exposure or the toxicity testing itself. So una nating i-prepare ang ating plant extract. Hindi na po yung plant material, yung extract na po ang kukunin natin from the plant. So you would need the plant material. It would either be a leaf, a stem, or a root. And you would need mortar and pestle kung meron ka, or pwede na ang knife, pwede na ang scissors. Pinakamabilis pong gamitin, blender. And you have a beaker and the solvent that we're going to use for extraction. And we'd make use of a water bath kung wala po tayong rotavap or rotary evaporator. So for the simple extraction, ito po ang medyo matagal. I-air dry po natin ang ating plant material or oven dry. But I would suggest na air dry po ang gawin natin. Bakit? Pag oven dry po kasi, magsasama tayo ng mataas na temperature, po pwedeng meron pong masira na mga compounds at very high temperatures. So it's best that we air dry it for 4 to 7 days. Yun nga lang po kasi, kung minsan, ang mga roots very thick or yung mga stems very thick na meron pang water content sa loob. That's the time na i-oven dry po natin yung material. And then you pound it. So ganito po ang kalalabasan niya. Talagang pinong-pino na siya. Now, what do we do next? Ibababad natin sa solvent. Very important po ang solvent natin for 24 hours. Iwan po natin siya. Just enough to cover lang po. Okay? So we could use either methanol, 90% ethanol, N-hexane, ethyl acetate, petroleum ether, carbon tetrachloride, dichloromethane, and acetone. Ito po ang mga suggestions ni Ahmed et al. At si Ogugu et al. Pero for brine shrimp lethality assay, I would suggest that you make use of this one, dimethyl sulfoxide. This is again suggested by Ahmed et al. At a different year, 2013. Kasi ang ating mga brine shrimp po, kaya nilang mag-thrive. dito sa dimethyl sulfoxide. Hindi po sila masyadong affected nito. Kaya mas magandang yan po ang gamitin natin. Kasi for us to test, then we should have the environment of the shrimp or the brine shrimp to be very, very close to its natural environment. And then after that, nababad na natin si solvent, natapos na ang 24 hours, nakita natin nag-change na ang color ng ating solvent, then gagawa tayo ng water bath. Ang water bath lang po is, eto may kaldero tayo, naglagay ka ng tubig sa loob, siya ang iinit mo. Ipatong mo lang etong beaker na meron yung ating extract. By the way, I forgot to mention, we have to filter. So from here, eto po, ganyan po ang itsura niya. Ano po? I-filter po natin siya para purong extract. Wala na po yung mga pira-pirasong leaves. Okay? And eto na po yung ating i-water bath. So ipapa-evaporate lang po natin yan. Pero wag din po nating hahayaang masunog. Dapat meron pong maiiwan yung minimal na content na nandun po. Water bath is one method. Pero ideally po sa laboratory, we have what we call as a rotary evaporator. So eto po ay parang malaking rice cooker pero ang laman po niya ay oil. Eto pong glass na ito, ang laman po niyan is yung plant extract. Umiikot po ito kaya siya rotary. Habang umiikot po ito, nag-e-evaporate yung solvent. Papunta po siya dito sa kabila na glass. Tapos ito ngayon ay magdidistill at babagsak, maiipon ulit dito sa kabilang glass. So kung ethanol ang ginamit mong solvent, po pwede ulit magamit yung ethanol na nandito. Naipon ulit siya. Unlike for water bath kasi, talagang ipapa-evaporate mo na yung solvent. So eto na po, naiwan na po ang ating extract. Now, the next thing that we do is to hatch our brine shrimp. So, ang brine shrimp po, easily available din. Kaya po ito yung mga pinili kong mga studies kasi easily available tong mga ito. Mabibili lang po ito sa mga pet stores. Ito yung pinapakain sa fish na nasa aquarium. Yung parang maliliit po na na mga eggs. Oo, hindi po yun pellets. Yun po ay brine shrimp. So, you have the brine shrimp that you bought from the pet store and you make use of tap water. Okay lang ang tap water. Basta please make sure that wala po siyang content na chlorine. Okay? Distilled water, pwede na rin po. Sa salt, any salt will do. Wag lang po iodized. Okay? And then we have just a pinch of baking soda. Pwede tayong gumamit ng air pump. And a straw, kung wala po tayong air pump, yun nga lang po, hipan-hipan ninyo. Medyo matrabaho nga lang po. Or, and sorry, we have a pipette. We can make use of a pipette. And the measuring spoon, of course. A light source, probably a flashlight or a lampshade will do. And one big clear container na gagamitin natin para sa hatching medium natin. And the small clear containers. Pwedeng Petri dish, pwedeng test tube, pwedeng plastic cups. Nung ginawa po namin to, ang ginamit po namin na plastic cups ay yung lalagyan ng ketchup sa McDo. Kaya madali lang din po siyang i-acquire. So ang gagawin natin, unang-una, hatching medium. Paghaluin ang water, 1.5 teaspoon or 9 grams na salt, at saka pinch of baking soda para ma-neutralize lang po yung ating water. Okay? Ay yung hatching medium natin. And from there, so kung 1 liter yan, then you separate at least 100 ml. Etong 100 ml na to gagamitin natin later for toxicity testing. Ipag-hatch muna natin yung ating brine shrimp. And then, i-aerate natin siya. Eto, kaya ang ating air pump nandito sa ilalim. Kasi dapat ilalim yung ating air pump para magbabubbles siya pataas. Mag-incorporate siya ng air dito sa may bandang taas. Or kung straw ang meron ka, lagyan mo ng straw, wag mo nga lang hihipan. Diba? Ay, wag mong iinumin, hipan mo lang. Oo. And then, you put your brine shrimp eggs and then mix it. Tutukan mo siya ngayon ng light source or lampshade, pwede na. Tapos overnight yan. 24 hours, mag-hatch na siya. Okay? Now, you will not harvest the nuclei. Nuclei po ang tawag dun sa larvae nila. You will not harvest the nuclei immediately after 24 hours. Hayaan mo din muna silang mag-grow at maging adult. So, you will harvest for the next 24 hours. How do you harvest? Turn off the light, turn off the air, okay? Magsiseparate yan. Etong nandito sa ibabaw na itim, eto na yung mga empty na eggshells niya. Tapos yung mga lumulutang-lutang dito, this would already be the brine shrimp. Now, if your bottle is open mouth, then pwedeng kutsarahin na itong empty na eggshells para ma-harvest natin yung ating brine shrimp. Pero kung talagang masikip ang ating bottle opening, pwede kang gumamit ng pipette. Okay? So, i-separate mo na ngayon. Ito yung isang container mo, 10 ang nilagay mo dyan, another container, 10 again, another container, 10 again, another container, 10. So, you have 10 brine shrimps in each of these small containers. Okay? Separate na natin siya para minsanan ang ating exposure. Now, we go to the toxicity testing or the exposure. What we'll have to do here is to prepare serial dilutions. Okay? Prepare muna tayo ng serial dilutions bago natin i-transfer ang brine shrimp. So, what do we do? Apat lang po yung nilagay ko dito. For example lang po ito. Pero kunwari kung 10 ang inyong um, ang nandun sa inyong plano for your study, then okay lang po, walang problema. So, each of them, you have a separate container for each. Maglagay ko ulit ng 10 ml ng hatching solution, 10 ml, 10 ml, 10 ml. Okay? Now, for the plant extract, eto na ang serial dilutions natin. Kasi yung unang-una, dapat wala kang ilalagay na plant extract. That will serve as your positive control. Okay? That's your positive control. So, ibig sabihin, after 24 hours, dapat meron ka pa ring buhay na 10 brine shrimp. Okay? Pero for the others, eto na ang serial dilution. 0.25 ml ang ilalagay mo kunwari sa isa, doblehin mo yan. So, naging 0.5 ml. Yung next naman is 0.75. Let's say yung next is 1 ml. And then yung next, let's say, is 1.25. And then yung next, let's say, is 1.5. Okay? That is your serial dilutions. Okay? And then for pagka yung nagawa mo na yung serial dilutions mo, ibuhos mo na ngayon yung brine shrimp mo doon. So put 10 in each of those. And again, you wait for 24 hours. May I also stress pala that the brine shrimp would only live for 3 days. Kaya you should do your study in 3 days from the time na na-hatch ang inyong brine shrimp. Okay, so after 24 hours, this is what we'll do. We are going to take note of the LC50 or that is the lethal concentration 50% dead. Yun po ang ibig sabihin nun. So anong gagawin natin? Magka-count po tayo ng buhay na brine shrimp. Okay? Tititigan po natin sila isa-isa. Siguro mga 5 seconds na talagang hindi gumalaw, patay na po yun. Okay? So you're going to count the dead brine shrimps. Okay? So pagka ganito ang ating result, positive control to, 0 din. Nako, delikado. Ulitin ang buong experiment. Kasi ibig sabihin, pag nagkamali dito, ibig sabihin, hindi reliable ang iyong experiment. Okay? Pero for the LC50, 50% dead, 50% of 10 is 5, right? So, eto kung saan lumabas ang 5. Eto na ang ating LC50. That would be 0.25 ml. That's our LC50. Okay? Now, what if you have Sorry, dito pala dapat yon. Pag ang brine shrimps mo, dead ang 10. Dito sa positive control, dead ang 10. Dito mo siya uulitin. Okay? Pero yung kanina, since walang dead, tama yung results natin. Okay? Sorry. Now here naman, kunwari, ang dead natin dito ay 0. So tama tayo dyan. And then, lahat naman na dead. From 0.25 ml onwards, lahat naman na dead. Ibig sabihin, yung plant extract natin ay matapang. So you have to lessen the concentration. How do you lessen it? From 25 ml, go down. Okay? So 0.25 ito, babaan natin. Yung serial dilution natin magiging 0.125, magiging 0.0625, etc. etc. Okay? So kung gusto ninyong 10 ang serial dilution ninyo, wala namang problema. Basta as long as you have the concentrations to be exact for your serial dilution. How about naman if ganito ang results natin? So wala pang namamatay, 0.75 na ang pinakamataas. Ibig sabihin, mahina yung plant extract. So taasan mo naman ito ngayon. Mag-start ka, so 0.75 yan, pwedeng gumamit ka ng 1 ml. Tapos 1.25. Yun na yung mga kasunod. You go up. You increase the concentration. Okay? This would be articles that I have included here para po pwede po nating gayahin yung mga ginawa nila or po pwede nating i-pattern yung study natin sa mga ito. Okay? So, if you have a plant, it's actually best that you already have a plant in mind. That's the first thing. You could have, as I have mentioned a while back, an umbrella study on this. You could already study the stomates and the trichomes. And then you could already study the fungal endophytes and do a lethality assay. Kung meron kang 4 na groups, 4 na studies na to. And this would be one single plant. Put all your results together. Pagandahin ang papel. And then you could actually publish. Okay? And that would be a very, very big contribution already for us. Lalo na sa atin dito sa Pilipinas. Kailangan po kasi natin ito eh. We need to progress. And for us to progress, the key is actually with the science teachers. Which is why I leave you with this. The most conspicuous practical effect of science is that it makes possible the contriving of things that enrich life. That is according to Albert Einstein. Thank you very much, teachers. I hope I was able to share with you some things that you could use and include in your arsenal as teachers. Kailangang marami po tayong bala as teachers. Thank you so much po. Ma'am Cynthia, Ma'am April.
Very much. Thank you very much, Ma'am Madge, for
Thank you
that very
po.
clear explanation of that very technical topic.
Thank you very much,
Even
ma'am.
I able to understand clearly. Ang galing na pagkaka-explain. So at this
Salamat po.
point, we now open the floor for questions. You may
Sige
just
po.
type
You could
the question in the
type
chat
in your
box if
questions
you
at the
wish.
chat box.
There's one here. Kanina pa yata to. Is it okay daw for a plant? What's this? For a plant biologist, for a biologist, not a botanist, to validate plants.
As much as possible po, we would need po sana botanists to be the one to validate the plants. Mahirap po kasi pagka yung mga Actually, ang dami po kasi natin plants dito sa Pilipinas. Pagka may problema po tayo sa pag-identify ng plants, pwede po nating i-contact ang mga museums of natural history natin. We have one at UP Los Baños. We have the Museum of Natural History of UP Los Baños. You could email them. You could contact them in their website po. May I just go over the chat? Ay, thank you din po. Sana po magamit po ninyo siya. Wala na pong questions, mga teachers. Yun lang po. During the pandemic, students have limited
There's
movement.
one question, Ma'am Madge.
Yes po. Is it possible to conduct the endophyte isolation, be isolated in the house? As much as possible, I do not recommend that you do that inside the house po. Because sometimes the spores of the endophytes could actually fly anywhere. Madali po kasi silang ma-detach. Pwede pong mag-cause ito ng allergic reactions. We also have, saglit lang po ha, palakihin ko na lang po yung aking chat box. Are there variables related to the characteristics of the stomates like stress in the environment po? Yes. Yan po ang magandang i-discuss. Pagka yung nakuha nyo yung results ninyo, yan po yung mga isasama natin sa discussion. And then, can we have a copy of your slide, ma'am? Yes po. I think, di ba ma'am Cynthia, mailalagay po ito sa YouTube?
Actually, this is will be uploaded in YouTube, and then from there you can access. There's a replay of the presentation. You can access it through YouTube.
Yes po.
Just check out the STAR FB page again for that, for the link.
Yes po. Okay. Can we have, mukhang wala na po ma'am questions. Wala na pong questions, teachers? Kung kailangan po ninyo ng help, pwede nyo po akong i-contact. Meron naman po tayong FB. Hanapin nyo na lang po ako doon. Or kung i-email nyo po ako, pwede rin naman po, ibigay ko na lang din po yung email ko towards the end. Or i-type ko na lang din po ito. moespiritu1@up.edu.ph. Okay, that's my email address if you need to contact me for any questions po. Okay?
Okay. So with that, so the email address is moespiritu1@up.edu.ph.
Yes po.
So on that note, I would like to thank again, once again, Ma'am Madge for that very informative. Oh, but there's one na humahabol.
Ay,
Humahabol na question.
what's the difference between LC50 and LD50? Lethal concentration 50 and lethal dose 50. Sa lethal concentration 50 po ang ginamit natin for brine shrimp culture kasi sinubmerge po natin yung brine shrimp doon. Pag lethal dose 50, eto po kasi is yung ini-inject or ini-injection, pinapainom. Kaya po dose. Yun. Kung ano po yung number din ng organisms na 50% mamamatay, yun naman po ang lethal dose 50. Okay na po. Meron pa po ba? Can we use other procedures for brine shrimp culture? Pwede naman po, ma'am. Sir, yes, you could make use of other procedures. Pero basically po, the protocol is very similar because it's very simple lang po. Kaya halos pare-pareho lang po siya. Meron pa po? Sige, isip-isip po tayo. Baka meron pa po tayong pwedeng itanong.
So we'll give them a few seconds.
Sige po.
For more questions.
Sana po may ma-discover tayong mga plants natin. I
Pero
tried
pa ma'am dyan
brine
may
shrimp
comment.
culture pero namatay lahat. What would be the possible reasons? One possible reason na namatay yung brine shrimp culture mo, baka ang ginamit mo is water, tap water, fresh water. Nagsusurvive lang po ang brine shrimp natin ng 1 hour sa water, fresh water. Which is why, kung natatandaan ninyo, ang gagamitin natin is hatching medium plus yung plant extract. Or it is possible also na kung namatay silang lahat, talagang mataas yung yung effect ng iyong plant extract. So you have to bawas yung ating cereal dilutions. In water bath and rotavap, will there be a difference in the concentration of the crude extract? Wala naman po kasi tatanggalin lang niya yung solvent. Basta ang importante po is mag-iwan ka nung extract na talagang ano pa siya, nandun, hindi pa siya nag-solidify sa ilalim ng lalagyan mo. Kasi pagka pina-evaporate mo na lahat yun, magsusolidify na siya sa ilalim. So mag-iwan ka ng konting solvent doon para medyo liquid pa siya, magagamit mo pa. In brine shrimp lethality assay, a prerequisite po to test involving vertebrate animals. Okay. Yeah, it is a prerequisite. Pero I really do not suggest testing vertebrate animals when it comes to the level of high school, college, and even grad school. I would suggest that vertebrate animals would only be done in those that would have an animal facility. Actually, kailangan mo ng veterinary dito dahil yung pag-acclimate pa lang ng mga animals, importante na yun. E di mas lalo pa kaya kung maggagavaj ka ng animals. Mahirap kasi siya. Ang daming mga protocols niyan, ang daming mga permits na kailangan mong ipa-approve muna before you go for vertebrate animal testing. What solvent should be used to dilute the extract? Is this for the brine shrimp culture? I have given a list of the solvents to be used. Pero, basta wag lang yung sobrang tapang kasi po pwedeng magkaroon din to ng effect dun sa plant extract natin na po pwedeng ma-denature o masira yung ibang protein components niya. Kaya mas magandang kung ano lang yung binigay ko dun sa listahan, you please just refer to that na lang. Then from the results of the lethality assay, are we only limited to use LC50 concentrations as the maximum concentration? Not really. You could use LC75 if you would want to. Kasi kadalasan pagka cytotoxicity tests, yung pang-test nila ng posibleng pang-gamot ng cancer, marami sa kanila gumagamit is LC75. What I mean is the solvent to add with the extract before adding to brine shrimp. Ano po yun, ma'am? Solvent to add with the extract. Ayun na siya, yung nandun din sa listahan. Yun din yung gagamitin natin. Okay? I think wala na po.
Wala na. Yeah. I think all the concerns, questions have been sufficiently addressed already. So once again, ma'am, thank you very much from the bottom of our hearts.
Thank
Sa
you din po.
uulitin,
Yes,
I
ma'am.
hope you don't get
I'd be
tired of
honored to.
yes, to our request. So
Yes po.
I'd like to turn over again to our very beautiful participants and handsome participants. Thank you all for participating. And of course, thank you very much, Ma'am Madge, for this spending your afternoon with us. We hope to see you again, all teachers, in our future webinars. We still have 3 coming up until December. So magandang hapon sa ating lahat. Stay safe and healthy. Goodbye.